So I went to Maryland at the invitation of MedImmune back in the spring to see what the team were doing there and to meet with Pascal Soriot again and to meet with Bahija, the head of MedImmune and vice president of AstraZeneca. And I'm sure we're all aware, there's some incredible work going on there, and it felt entirely appropriate and fitting that we should invite Bahija to come and be our opening keynote address for On Helix. So without further ado, I shall hand over to Bahija with a very warm welcome. Thank you.
Thank you, Harriet, and good morning, everyone. I'm really delighted to be here today, and especially to talk about a subject that's really close to my heart. It's personalized healthcare. And I do believe that we are in a seminal moment. So I will talk to you about where we are. I'll give 2 examples and then look a little bit with you into the future. So I've been in this area for a long time, and you hear all kinds of definitions: personalized healthcare, translational medicines, and also I thought I'm going to look at what's the latest definition. And what caught my eye is really this one. It's a discipline with biomedical and public health research that aims to improve the health of individuals. And that's really the important thing, and that's why we come to work every single day. So translational medicine, definitely, in the last 20 years I've been in drug development, had absolutely transformed or shifted the paradigm of drug development. So this is how we used to do it. We'll identify a target, and we'll go through a very linear process, one step at a time, sometimes one department handing over to the other. 10 or 13 years later, 1.4 or more billion dollars later, if you're successful, you will get a drug that actually helps 30% of the patients. That was a success. You can even push it sometimes 30 to 50%, and that's really hugely successful. But the issue is, we didn't know how to identify the 50% that are actually going to get just the risks, but not the benefits. And I'm pretty sure you're familiar sometimes, for me on the headache, I can take Advil. Tylenol doesn't work for me. And that's okay if it's a pain medicine for headaches, but imagine if it's a drug for cancer. We are talking life and death. So it really behooved us to go and do things a little bit differently and try to identify a priori the patients that are most likely to benefit. So it meant we had to change the way we do drug development. Instead of going in a very linear process, is having a more integrated process that you actually go with hypotheses into the clinic to test those hypotheses and bring the learning back from the clinic into the lab. It was definitely true for oncology. That's where, in cancer, that's the most advanced application of translational medicine or personalized healthcare. So the shift we've seen in oncology is that no longer you look at the cancer as which organ it came from, basically it emanated from. Is it lung or breast or something like that? So when a patient comes, it's not any longer you have a non-small cell lung cancer, but instead you have an EGF receptor positive, ALK positive, non-small cell lung cancer. But it still leaves 42% of non-small cell lung cancer in this case that we don't know the molecular, at the molecular level what kind of cancer it is. But I'm happy to share with you that at least there is real revolution happening in cancer today, which we're no longer just focusing on the tumor, but also on the immune system and harnessing the power of the immune system to attack cancer. And that opens up a whole other area where, again, we need to do more translational medicine. We need to understand more the immune fitness of every single patient. So all this learning in cancer, when I came to MedImmune many years ago, with my background in cancer, the first thing the first project I tackled was outside of cancer. And that's really with the idea, how can we take this learning from cancer into other therapeutic areas? So the first project I worked on with my team was in lupus. So I learned a lot. I learned that lupus is an absolutely debilitating disease. It actually affects women, mostly at their 30s or 40s. And few months ago, or just a month or two ago, we had a patient who came. She was 23 years old. She was diagnosed when she was 19. And she already at 23, she had 2 hip replacements, and she was going to have a shoulder replacement. And that's actually not from the lupus itself, or indirectly. It's because the steroids, you have to take pretty high doses of steroids that completely damaged her joints. So it really behooved us to do something different. We know that there was one drug that was approved 2 years or 4 years ago in lupus. But again, it helps only 20% or 25% maximum of the patients. And the problem, what the doctors tell you, is I cannot tell if a patient is walking in, if they're going to benefit or not. That's 20% of the maximum of the patients. So we had to do things a little bit differently. So we and others in academia was all reported that type 1 interferons are very important in lupus. So we studied that internally, and we went back to the lab, looked at how we can identify. It's not easy to identify interferon alpha as a protein in the serum. So we actually looked at the molecular signature, a gene signature. So we identified that. That's what you see in the top panel. So the red ones are normal individual or patients that have high interferon. So we went, then looked at lupus patients. And what you can see in the bottom panel, the green ones are normal people. You can see they don't have a high interferon signature. However, when you go into the patients, you can see that not all the patients have these reds. You can see 2/3 of the patients have a upregulation of the type 1 interferon pathway, while others, they must have other pathways that are activated, but not type 1 interferon. So we went with this data into the clinic in phase 1 and in phase 2B to try to look, are these patients, the red patients, going to benefit and the green are not going to benefit from the type 1 interferon. And as a matter of fact, this is data, just selective data that I took from the phase 2B. And what you can see, the gray panel on my right, tell you women have problem direction here, but it's me probably. So you can see that the gray it's a placebo, and you can see that when you have on this panel here, when you have a high interferon alpha pathway activated or type 1 interferon alpha activated, you can see that you actually benefit as in lupus patient. This is one endpoint, the SRI. However, on the other panel, what you can see that if you have a low signature of the type 1 interferon, you don't do better than placebo. And that was really important to have this data before you even go into phase 3. How does it manifest for the patients? This is, again, a patient that has a high type 1 interferon. You can see for the skin manifestation, and that's one you've seen on the face and on the body. If you have a high type 1 interferon, you completely have a complete skin clearance. So right now we are testing these. We dosed the first 2 patients in phase 3. But the most important thing we will have, if we are successful, we will have this drug getting into the market with the diagnostic that the people, the green ones that you've seen, don't have to take this therapy. And then taking that learning from oncology, again, into this autoimmune, and we said, okay, if we if we know type 1 interferon is very important in SLE, can we look at other autoimmune diseases and say where the type 1 interferons are going to be also important? And this is what we've done first. So you look at, again, you have the normal patients or the normal people. You have the SLE patients, you see the same things. When you have a high type 1 interferon, you're red. You have some SLE patients that have low interferon. But again, if you go to polymyositis, dermatomyositis, or IBM, or even RA, you can see a subset of patients that look a lot more like the SLE patients, right? So they have a high type 1 interferon. So with the simple blood test, if we're successful, if we get it past the phase 3, that you can imagine a future where if you walk into the doctor's office with an autoimmune diseases, instead of just giving steroids, you'll be first tested. Do you have a type 1 interferon? So it's that kind of learning and hopefully coming from the oncology into autoimmune diseases where we can give other alternatives than just steroids. So this is another just quick example in asthma. So you know you're all familiar with asthma, but what I really want to talk about very quickly is the severe asthmatic. So these severe asthmatic are not the ones that, these are patients that actually failed all corticosteroids. Nothing is working for them. There's absolutely nothing. And they actually live from one exacerbation to the other. So when they get an attack, an asthma attack, they end up in the ER. And one asthma attack could be their last one, with the difficulties to breathe and everything. So we looked into that population with severe asthmatic. If you look at the biology, this is not meant to go through the biology, but just to tell you that there is more than just one pathway, and there is a huge heterogeneity within that population. So we set out to look for, first in that population, to characterize the differences. So to cut the long story short, we find 70% of these severe asthmatic have very high eosinophilic, so we call them the eosinophilic asthmatic. Another population that's more virus susceptible, I call them, so we call them the Th2 asthmatics, which is more on the inflammation. And then you have another subpopulation, which is the atopic asthmatic, and those are the ones that receive the IgE. So going with that learning, we zoomed in on what's really important in each pathway. And we actually developed a product, benralizumab, for the eosinophilic asthmatic, tralokinumab for the Th2, and then we have TSLP is another molecule basically that's also going through the testing. The two first ones are in phase 3, but the most important thing is for each these molecules, we will have just a blood test. Though if you present yourself and you are actually a severe asthmatic, first you determine, are you eosinophilic, are you a Th2, are you on the TSLP, and then hopefully all with this blood test that you can get the right medicines in that critical moments where you're in the emergency room. So this is, it sounds really simple, right? So in all these cases, we are lucky, and if we're really lucky, we will have a success in phase 3, but we have the diagnostic developed directly with the therapeutic, and all these diagnostics have been are being tested in phase 3. But I don't want to leave you with the idea that this is really simple. It is a lot more complex, and especially when you have to actually bring the diagnostic into phase 3, you have to have identified it in preclinical testing, right? So you have to get into phase 1 with the first testing, then there is a lot of the validation and everything until you get into phase 3. So that's why it is really important we can't do it by ourselves. You have to collaborate. You have to collaborate with academia. You have to collaborate with other companies. And in this case, especially when we get into the whole diagnostic. We chose not to do it in-house, but to also collaborate with people who know how to do diagnostic, which is the diagnostic companies. So there's definitely more challenges, even in oncology, because you move from sometimes, most of the times, if you have successful compounds, you move from phase 1 to phase 3. You don't even have the time, to test the diagnostic in a proper way. And you've seen several times where you get into the market and then go back to do some studies on the diagnostic. So the challenge is there. And but we have, we're still not in all the areas that we should be, just because sometimes it's the science, sometimes it's our knowledge and the difficulties of finding these biomarkers or diagnostic. But I would like, with all these challenges, I think it's still really exciting and looking back where we were and where we are today. So I just, in my last 5 minutes, just look into the future. I think we made a huge progress looking at the personalized healthcare, but how can we now, the technology I think is pushing us there in a good way to look into individualized treatment. So, and for that I think we have to look really beyond our industry. So look at the explosion of technologies that are happening right now. So yes, most of you, probably some of you have the Fitbit, and they're measuring how we walk, how much we walk or we sit or something like that. But it really is a great opportunity for us now how to use these technologies to actually look at more of a continuum, not just a specific biomarker, but actually for every single patient measure. For instance, that asthmatic, when you're going to get an attack, can you measure before you get the attack, when you're taking the drug, after you took the drug, and actually try to understand that even more. And that's what's called the whole big data. We can actually look at not a single biomarker, but a continuum of parameters. And I think now with all this big data and digital health, this is going to be possible, but this really behooves us how we bring it even to the drug development, even before, and learn from it. So I just leave you with this one, that we definitely, if we look into the future, we're going from personalized healthcare to more individualized treatment. And I think that's really the vision. That's not impossible when we look how things are going, but it takes us to be even more imaginative and go outside of our industry. And only if we do that we actually can now, I just want to leave you with this thought, start actually looking at how we can ask for pay for value. And I want to leave you with this thought, because now with these technologies, we actually, when you say pay for value, is great for the economy, it's great for everybody. But now I think we will have the tools to do that, which is looking at the compliance, and we can have that in the discussion. And if we do that, it's going to be good for the health system, and most importantly, it's going to be for patients. Thank you.